I am a professor here at Western in the biology department, and we have a mix of biology students and Huxley students and maybe some other students in the room. And so thanks for being here. I'm going to talk today about the Living Snow Project, and this is a program that I'm calling community-enabled science. Oftentimes it's referred to as citizen science. The reason we're transitioning out of that name is because anybody can participate. It's not dependent on whether you are a citizen or not. So community-enabled science is how I'm going to refer to this. And it is a program that engages the outdoor recreation community as citizen scientists in research exploring the biodiversity of alpine snow ecosystems. By empowering people of all ages to participate in science in the places they love to play, we help to cultivate a deeper awareness of our living planet and a greater appreciation for the conservation of threatened ecosystems. So that's kind of the mission statement for the Living Snow Project. And today I'm going to do, it's kind of a focal point of the research that I do in my lab. So I'm going to talk to you about the research that I do on alpine microbe community biodiversity. So that is going to get into some of the technical details of the science that I do. I'm going to talk to you a little bit about a project that I'm doing, which is a social science project that's studying how participating in community-enabled science impacts the volunteers and how they feel about conservation. And then I'm going to talk a little bit about program development. So how does a scientist like myself go and start a program like this? How do I develop a program like this? This isn't what, not every scientist decides to engage in the community in quite this way. So I'm going to talk about the process that I've gone through to develop the Living Snow Project, and then the different outreach components that include some other collaborations outside of just my lab here at Western. And so when we're studying alpine snow, how many of you have seen the snow turn pink before in the springtime? Hands up high. Like, I would say 75% of you have seen this. It's pretty exciting. It's surprised snow algae are what turn the snow pink, so there are these algae living in the snow. And it's never ceases to surprise me how into pink snow people are. When I tell them that that's what I study, they're like, oh yeah, I've seen the snow turn pink. People get really excited. More excited than, say, algae that live in the ocean, which I also study. So pink snow can be quite popular. And so what are snow algae? They are a group of different organisms. So if you read Wikipedia, Wikipedia will say it's Chlamydomonas nivalis, which is this one species up here. Robin Matthews from Environmental Science took that photo. And so she's been looking at these groups for a long time as well. They're closely related to freshwater algae, and some soil algae. So it looks like they made the transition into snow from freshwater or soil. But it's not just this one species. There's in fact lots of different species of algae that can live in the snow. And they don't all look red. Sometimes they can look orange or sometimes yellowish. And they can turn the snow different kinds of colors, but typically you see it as pink. And it can present on the snow in a bunch of different ways. So it can look like these sun cups here from the Alpine Lakes Wilderness in central Washington. And you can see that there's patchy, and the algae are kind of growing in layers, and they are helping melt the snow there, which is creating a little pocket or a sun cup. This is from Glacier Peak, which is also in central Washington, and you get algae kind of growing well distributed on the snow surface. This is a picture of the Sisters Glacier, which is just right in our backyard, like right between us and Mount Baker. When you look at this glacier, you can't necessarily see the pink from this angle, but if you get up really close, it's light pink, and there's a bunch of snow algae living there. They just don't make the snow bright pink. You can get different appearances of the snow turning pink depending on what side of a mountain you're on. So this is a Goody Mountain, and I climbed up and over this mountain, so I collected on one side and then the other side. And on one side we had icy snow, and we get this dark patches that appear on the icy snow, and on the other side we had very slushy snow, and the pink appeared in runnels. So even geographically close places can host different communities that look different on the snow. You can have them on glaciers. You can also find them on leftover little snow patches. Easy Pass is also in our backyard, right in the Cascades. And there's little patches of pink on that little leftover snow patch in August. We see it on glaciers. This is a place where snow and ice exist year-round, but we can also find it in annual snowpack. So this is places where we have deep snowpack, like adjacent to the Mount Baker ski area, for example, which is one of the places where I have an intensive study, where the snow will melt away. So that's this basin right here. The snow in this basin will completely go away by the end of the summer. So we can look at it under the microscope to characterize it. We can look at it on the surface of the snow, and we can see it presents in all these different ways. And it's important in the environment of alpine snow ecosystems, and really important also in polar systems, because snow algae can change the albedo of snow, which is the reflectance of the sunlight on snow. So when you have white snow, it has what we call high albedo, and most of that solar radiation that's coming from the sun is reflected off the snow surface. When you have something that's making the snow dark, that can be dust, that can be black carbon, that could be algae growing in the snow. It darkens the snow surface, and it absorbs far more solar radiation than if the snow was white. And so this article was written a couple years ago in the New Yorker, and it has a kind of exciting title, Why the Last Snow on Earth May Be Red, because there's been a number of studies that have come out just in the past couple of years that have quantified the degree to which algae can change the albedo of the snow. And when algae are blooming, when they're present in their bright red, they can increase the rate of snow melt by up to 20%, which is a pretty big deal, especially for glaciers, which are already threatened because of warming climate. So, despite the fact that they're pretty important, snow algae and the groups of microbes that live with them are fairly understudied, which means there hasn't been a lot of scientists who have been working on these. For example, when I'm looking in the Cascades, I find new species all the time, because the last papers that were publishing snow algae, or publishing about snow algae in our region were from the 70s, and they were papers that don't include using DNA technologies up until like the last few years. So this is a fairly understudied system. And so there's a lot of things that we need to learn about them to understand how they're impacting a system. And snow algae have complex life cycles. So one of the first things we need to know is who's there, so which species, but we also need to know how do they get around, how do they repopulate the snow. And one of the theories, or one of the hypotheses, is that in the summer, they produce a red layer on the surface of the snow. And then in the fall and winter, we get new snow falling on top of that, and the algae that are there turn into cysts. So they kind of suspend metabolism, and they overwinter underneath that snowpack. So if this was a glacier, this would be on a snow or ice surface. And if this was a place where there's annual snowpack, this would be on rocks or soil surfaces. And then in the spring, when the snowpack is melting, and it is what we call isothermal, or completely melted throughout the whole snowpack, those cysts from the year before can undergo developmental change and turn into swimming cells, because algae have complex life cycles, and they have a stage in the life cycle that swims. So it turns from a cyst to a swimmer, and then they can swim up the snowpack and multiply, and then reestablish as photosynthetic pink cysts. Okay, so this is one of the hypotheses about how they repopulate the snow. So once you have a population of snow algae, they can come back from year to year. We have some evidence of that. These are some images of algae that I took from my field microscope. So I'm going to play this video, and it's going to shake a little bit. Oops. Oh, geez. I'm hopefully going to play this video for you. There we go. There we go. So I was able to capture a swimming cell along with these photosynthetic cysts. When I say photosynthetic cysts, it means they're still doing photosynthesis, even though you see them as red. The reason is because the chlorophyll that's green is underneath the red pigment. So it's kind of the opposite of what happens to leaves in the fall. When the chlorophyll goes away, then the leaf turns red. before senescence. This is the other way around. And so the dark red pigment is the dominant pigment when the algae is growing and moving. So we were able to capture swimming cells, and this is in early June, so this is when the populations were just establishing. And we were also able to capture some of these cysts still swimming. So they actually lose their flagella, the swimming cells lose their flagella before they establish as these photosynthetic cysts. And so we have good evidence that suggests that this mode of reproduction is happening. But we also suspect that algae may be wind dispersed, or those spores can also blow around on the wind and can re-establish populations in other places. And so we're interested in differentiating between these two modes of dispersal based on the data that we're collecting. The species also develop, so when we collect them in the field, they can be at various stages of development. So these cysts are not static, so this would be what we would call an immature cyst, and develops into a more mature cyst. This particular species, we don't actually know if this is these are two separate species or two different stages of the same developmental stage. There's a lot of basic biology that we're still learning about these communities. They also live together, so you don't usually find just one species. Even though you pick up, there's a patch of pink here and a patch of pink over here, they can have different groups of species that are living together. And so here's some scanning electron micrographs showing a set of different species. And then here's a time series, 3 different weeks from last summer. And each of these were collected from the same place. Okay, so we can't even go back to the same place in successive weeks and see the exact same thing. So these communities are really dynamic, developmentally, reproductively, and in terms of their communities. And the community function is really important because they don't live by themselves. And so if we think about this as a alpine snow ecosystem, there's a lot of things, there's plants, there's predators, there's scavengers, there's herbivores, but inside this little pink patch of snow here, we have algae, bacteria, and fungi all living together. Okay, so I was talking to you about the algae, but they also live in partnership with all those other things. Okay, so that is all the stuff that you need to know as I launch into talking about what we are interested in terms of research. So studying the alpine community biodiversity. And I want to thank first, before I get into the data, the people who made this possible, because there's a bunch of Western students who have worked on these projects. And so I'm just going to flash this up here, and I want to make particular mention to Rachel Mallon, who was a master's student in my lab, and we'll be talking a lot about her master's thesis data today. There's also a couple of these people in the room, Jack and Anna, and maybe Ruth, maybe? Back there. Okay, great. So a lot of Western students have contributed to this research project. And so as we think about the interactions between these groups, and I just mentioned that they live in communities, we have big microeukaryotes, little microeukaryotes, bacteria and viruses, and tiny microeukaryotes. All these organisms are interacting within these pink patches. And we need to understand their diversity and their community structure so that we can understand their ecology, so that we ultimately can understand impact. So hopefully that makes that you all agree with me. So our first step is to understand the diversity of these organisms. Then we understand how they interact with each other. We can learn about how they disperse, and then we can understand how they're impacting snowmelt and glacial and snow ecosystems in our region. And so because these communities are complex, the ways in which we have chosen to study this is using DNA. Because it can be really difficult to identify these species, right? Sometimes they just look like little red balls, and sometimes 2 little red balls can be different species even though they look the same. And sometimes the same species can look different, like that developmental sequence I showed you before, if you catch it at different times in the life cycle. And so people who study microbes often use DNA as a better measure for being accurate about measuring community diversity. And so what we do is we take a sample of pink snow, which is a mixed population. We extract all the DNA out of those samples. we sequence a small piece of DNA. This is about 400 base pairs, so it's just a tiny piece of DNA, and we sequence millions of these tiny pieces of DNA. So we could have up to 100,000 sequences per sample. And each of the samples has a community fingerprint of all of the organisms that are living there. So we have a couple of major research questions. We're interested in what snow algae species are present in alpine snow across the Cascades. Are there distinct snow algae assemblages? So when we see these organisms living together, is that a consistent relationship? Do they have biogeographic signals? So are populations that are close together geographically more similar than populations that are farther apart? And do these communities have a seasonal succession? So is it the same, or do they change over the season? And I give you a little bit of a preview to that by showing the 3 weeks from last summer, where we saw that they did change over time. But we wanted to see if we can define that change over time. So to answer these questions, we designed a study where we are looking over large geographic scales, sampling across the mountain range. Okay, so across the Cascades, that's 1,000 kilometers. We also were interested in being able to look over at a much more detailed place where we could go and sample regularly, and so we do sample right up Highway 542, right next to the Mount Baker ski area, where we can go every week and do a fine scale study. And then we can look at both annual temporal variability, so we sample from year to year, so I can tell you if 2017 was similar to 2018, is similar to 2019. But then we can also look at seasonal changes, so we can say is early in the summer season the same or different than later in the summer season. And in order to do this is a lot of sampling, especially over the mountain range sampling. That's where the citizen scientists come in. That's where the volunteers come in. Because I and my students cannot go everywhere in the Cascades, and we've just expanded our sampling from Washington to include Oregon, British Columbia, California, and then we're starting to expand out of the Cascades into Montana, Wyoming, and Idaho. So in order to get a lot of samples over this large geographic range, we recruit folks to help us collect samples when they're going out into the mountains. And there's a lot of people going out into the mountains all the time. And so they help us by getting a sample kit. So we send folks a kit. They scoop pink snow into a tube. That tube is filled with RNA later, which is a preservative for nucleic acids. And so that will make the sample, that will preserve the sample at ambient temperatures for about 2 weeks. So it gives folks 2 weeks to get the sample into a refrigerator, which is plenty of time usually. And then it's okay in the fridge for a while. So then they can send it back to us at their convenience. And we get really excited when they send back kits. Jack and Anna in this room have spent a lot of time receiving these kits and entering the information into spreadsheets. This is a kind of heroic effort. There's just like one little picture here of kits, but we make hundreds of these kits in the springtime every year and then mail them out to hundreds of people. It's a huge effort. We also have places where people can pick up kits, and we'd like to do more of that in the future, but it also is hard to get people to go places and do things. It's much easier if you send it to them. So this is how we sort of set up the Living Snow Project, and people go and sample. So there's some videos here showing folks scooping snow into tubes. And basically anybody can do this, so if you're interested, we're always taking more volunteers, but you scoop the snow into the tube with the cap, you shake it, and everyone puts on a glove to keep everything sterile. And so far, we have seen really high quality with our samples, no indication that there's any systematic contamination from volunteers. So we have, my lab has been at this since 2013, but from 2013 to 2016, I was doing kind of a pilot study, so we just took a few samples. I had 40 samples from 18 unique sites. 2017, we grew a lot, and we got 210 samples from 38 unique sites. Then last year, I did some big advertising to recruit volunteers, and we had over 260 samples from 100 plus unique sites. And that's what's really important, because it would be impossible for me to visit that many unique sites in the summer. And then we are still doing our tallies for 2019 and still getting folks to send us samples. So I don't have the data from 2019 yet. But I do have some maps that Jack made, and so he, using the GIS coordinates, we can plot all of the different points. You can see how we've been expanding geographic range. The pink dots are the samples collected by my lab, and the green dots are samples collected by community members. So we sample directly into these tubes. This is, I don't want to go into the details of this, but any of you that are biologists, we sequence using aluminum MySeq sequencing. We do a bunch of different quality control with bioinformatics with our samples. We also do some microscopy, like microscopy and SEM. I already showed you some of those images. And we've been using GIS to get environmental and climate data. Okay, so the data I'm going to talk to you about for the next few minutes comes from 2017. And this made up Rachel Mallon's thesis. And we are just about to submit this paper, so we're pretty excited. Any day now. The scientists in the room know that feeling, the any day now submission. And this map shows these different localities or regions that we have samples for. And so this study includes 127 samples. We have learned a number of important things. I'm going to give you the punchlines first, and then I'm going to show you the data. So first, there are new species in our region, which we suspected, but we have evidence to show that. The pink snow microbes form assemblages that are consistent. Okay, so we can see that these different microbes live together, and they consistently live together, and we can define the assemblage groups. But they are not distributed geographically. Okay, so that means that the ones that are close together are not necessarily most similar. So we've learned that, in fact, they are more similar by elevation and by the time of collection or the time in the season that they live, not geography. And not only are they similar by the time of the season they're collected, but there is a seasonal succession where we can consistently see them change over time. So this is one of the results of the data. So if you see on this axis, it shows us all of the different species that or genera that we have pulled out of our millions of sequences. So we have 10 million sequences in this analysis. So this ends up being quite a big data exercise. And what you can tell from this is that we have a bunch of things that are rare or potentially endemic, meaning those are taxa that we don't find very often. And then there's only a few taxa that we call dominant or cosmopolitan that are in almost every sample. Okay, so that tells us a lot about the ways that these pink snow communities operate. There's a few taxa that are found everywhere, and there's a whole lot of things that are really rare. This is consistent with this hypothesis of a rare biosphere, which we see in other microbial communities. So it means that snow are similar to other microbial communities. And then when we look at the annotations by samples, so now sample is on this x-axis. The green part of the bar is algae, and the gray part of the bar is other. So those are the fungi and the other little micro-heterotrophic things that are eating the algae or eating the bacteria that live with the algae. And so about half of the cells, or half of the things in a pink snow patch are algae, and the other half are other things. So this is a robust community structure of algae and other things. We had 41 unique total taxa, and between 7 and 18 different kinds of algae per pink patch. Okay, so this is really different than if you read on Wikipedia and it says pink snow is climbing monocells. Right, that's way oversimplifying. That videos that I showed you of the swimming cells from Bagley Basin, that is in fact a new species. This is just to show you that we did some comparisons within our 400 base pairs of DNA. It's either 2 or 4 base pairs different than other described species that look the same. So there's Clinamonas is the genus, and there's a couple of species that exist in the database from Europe, and we're 4 base pairs different from that. In microbe DNA land, that's pretty different, so even though they look the same, we're really different from the European species. It looks like we're about 2 base pairs different from the species, similar species in New Zealand. And there's a bunch of other things in the database that are just called uncultured algae. So when I take my sequences from this big data set and I search against the databases, sometimes the best match I get is uncultured algae, which is not very informative. So there's a whole lot of other new species out there in snow for us to describe. So this is just one of them. We can also look at these. This is also the Bagley Basin, and this is a set of data that was taken from 2 different samples that were right next to each other. And some of the algae were shared between the patches, but each of those patches had unique sequences or unique algae. So there's quite a bit of unique diversity. The next thing we did is we took all of our samples and all of this DNA in each of the samples and annotated each of those pieces of DNA, so we gave it a name. And then we did a multivariate analysis called a principal components analysis and looked at the relationships between each of those samples. And what that does is it allows us to look at all of the, when you have 10,000 things in one sample and 50,000 things in another sample, how do you even start to do those comparisons, right? That's really challenging. If you're doing this with terrestrial plants, you might have like 3 species of trees and then 5 species of trees. And that comparison is something you could do more manually. So these multivariate analyses allows us to look at this complex data set all at once. And what we can see is when you look at this, that it looks like there's some clusters, right? Does that seem like it makes sense? Like there's some over here that look similar, and some over here that look to be grouping. And so we did an analysis called a k-means cluster analysis and used an elbow method for the statistics to tell us how many clusters within our set was most likely. And this suggested that we were going to have 5 clusters. So the elbow method is where we see the inflection point. And so 5 clusters, and we then coded these different clusters with shapes. And we had, so these are where those 5 clusters fall out. So we have squares, pluses, circles, squares, and X's. And again, a sample ends up wherever it ends up on this graph based on the different species, based on the different sequences of DNA that are in each of those samples. So if we simplify this and we just look for the middle, or what would be the average of each of these clusters, we see this. Okay, so this is just a simplification of the previous graph. And we have a cluster X and delta that are pretty similar. We have a cluster circle and square that are pretty similar, and then we have this cluster plus out here that's really different. Okay? So we can take each of these clusters that are built up of samples and look at the average community structure, the different species that are found within each of these cluster groups. And that's shown here in these pie graphs. So X and delta are here, plus is down here, and it turns out that this pink wedge is Chlamydomonas nivalis, so that is the most common, or the poster child for snow algae. These are totally different species. And then circle and square. We broke this down so that we could look at just the algae taxa. And here's some images of what these dominant groups are. So here's our new species, the Chlamydomonas, which is in this pink. We're not sure who that light pink is. Its annotation is uncultured Chloromonas, which could be anything. So it is something, it is one of these guys, but we're not sure who it is, because when we collect the samples, we take all of the DNA from the sample. And so we have to go back and do further studies, figure out exactly who that guy is. And then there's our Chlamydomonas nivalis. And in addition to looking at the algae, we can also look at the other taxa. There's less variability and less diversity within the other taxa. But I think that's probably has to do with the fact that there's been even less work done on those groups than on the snow algae, and so our databases are not very good. So if you're interested in doing a study where you can find new species, this is an excellent place to do that. Okay, so we've established that there's these communities, and they are different from each other, right? And they are not different from each other based on where we find them. because you can find two samples that are close together and they can be really different. So why are they different? What we did is we went and pulled a bunch of environmental data that we could get from the GIS coordinates from where each of the samples came from. Because remember, a lot of these samples are collected by volunteers. So besides having the sample, the only other information we have is the GPS coordinate. And so with those coordinates, we pulled information from a bunch of different environmental variables. So we looked at Julian day of sample collection, and so Julian day is just we number each of the days of the year from 0 to 365. We looked at latitude, longitude, the day of the date of the first frost-free period, mean annual precipitation, mean annual temperature, degree of continentality, which is basically how far from the coast you are in Washington, precipitation is snow, slope, elevation, aspect, etc. And we ran some statistics on these and found that this set of variables were different between the different assemblages. In a particular note were that elevation and time, which is that Julian day, seemed to predict the difference between our samples. And so you can see that this is elevation. There's basically a gradient of elevation change across the different assemblage groups, and there's also a change in an increasing increasing relationship between the day of collection and the different assemblage groups. And so if we look at the relationship between elevation and time on this axis, we can actually see that there is almost a linear relationship between these variables. And so when we look at them with relation to the different assemblage groups, what we're finding is, or how we're interpreting this, is that these two are early season, low elevation assemblages. And then we have some late season, higher elevation assemblages. And then this plus one is quite different. But what's really interesting is these early season, low elevation assemblages. So they're different species, they're less well-known species, and their habitat is very threatened. These are things that are found very low and in early, like even into May. Those are places in Washington where we're not going to have snow in a few decades. So we're not going to have consistent snowpack, and therefore these assemblage groups, and specifically those algae, are likely threatened. And we don't see them moving up in elevation as time goes on, right? My first hypothesis when I started this study back in 2013 is that everything would basically look like that one pie, or we would have one kind of snow algae and it would just keep moving up. Okay, so this is pretty, this idea of putting together an assemblage group of microbes and limiting them to a particular environment is a new idea. We also see that the seasonal succession is driven by the algae, not by the other taxa. And so this is another type of multivariate analysis where we looked at just the algae taxa and just the other taxa. And here we see the different groups. So we see the different assemblage groups. So we have plus, or X plus, X, excuse me, I can't speak, X delta plus, and then squared circle. And you see how there's they like move along and there's an overlap between the groups, but they don't all completely overlap. That's showing us that there's a succession, whereas these are the other taxa. See how all of those are just sitting on top of each other? It means that those other groups don't change over time as much as the algae change over time. And so algae are the primary producers of this ecosystem, and so they seem to be driving the changes that we see seasonally, which is not that unusual, and it's pretty consistent with what we know from other types of terrestrial ecosystems. The last thing is that we learned that the algae, that these communities are becoming more diverse through the season as well. And so we see that there's low diversity in those early season, low elevation groups, and there is more diversity, so more taxa. And we think that's because the longer the algae can live on the snow, the more sugar they produce in the snow, and the more things can live there. Okay, so they're creating more favorable habitat for other things. Okay. We also looked at things on a basin scale. I'm not going to spend a lot of time talking about this, but you can see this is all photos from 2017. You can see this environment change over time. April, we have really, really deep snowpack. By the time we get to September, there's very little snow. We have another study going in my lab right now because this year, 2019, we lost almost all the snow by mid-July. So it looked about like September 2017 in mid-July this year. And so we're looking to see, and that's just an ongoing project that is looking to see how that drastic change in environment impacts the community of snow algae. I am going to jump ahead a little bit because I want to leave time for questions, and I think that that we can move through some of that. So the last thing I'm going to leave you with some of these technical questions is we're asking now, what are the environmental conditions that select for this community structure in a more detailed way, especially when we're having dramatic changes in the environment. And so this ongoing work is being done in my lab by students at Western. I'm also collaborating with Ruth Sofield, who's in environmental science, and we're looking at the snow chemistry. So that not only are we pulling all of these climate variables from GIS data, but we're actually going and measuring the snow that is adjacent to where we are collecting to see how that type of environmental variable is selecting for the community. All right. So this project, a lot of the data was processed by students in my lab, but a lot of the samples are collected by volunteers. And we would not be able to do this large of a scale study without that. And so that's one of the reasons why I had the idea about having a community-enabled science program is because it 100% facilitated my research goals. It was also a really excellent way to reach out to the community. But in order to do that, I need to get people to volunteer, right? It's not always, even though people are excited about snow algae, I still had to do a lot of work to figure out how to recruit folks to help us with this work. And so I did that. I had to figure out how to communicate with volunteers, and I had to develop tools like an app, a website, Instagram, and all of this I did within partnerships that I developed, and then including some sponsorship. So ways in which we work to recruit volunteers within this project and communicate with volunteers, I did things like give public talks, like I'm doing now. We've developed social media presence and partnership with social media. So the Northwest Avalanche Center, which does all the avalanche forecasting for the Pacific Northwest, is a partner with us. And the first time they posted my call for volunteers, I had 100 people sign up within an hour. So working with community partners like that has significantly enhanced my presence in our community. And then I've developed partnerships with groups like the Mountaineers. Patagonia Seattle is a partner this year. Patagonia is helping us set up outreach events. We're going to hold one maybe in February this year. They also were a place to pick up and drop off kits for people in Seattle. The Mountaineers was also a place to pick up and drop off kits. They also have included participating in the Living Snow Project as a way for their participants to earn their conservation credits. So they all have to do conservation projects to earn their Mountaineer certificate, and participating in the Living Snow Project is one of those options. This past year, we developed an app, and so in addition to folks collecting samples for us, we want to go big and we want to get a lot of observations. So we've learned a ton from the samples that people have collected for us, and we're going to keep collecting those samples, and I want to collect them over decades, because in order to understand how climate change and changes in the environment are impacting the biology, we just have to keep sampling year after year after year, especially in snow environments that are very different from year to year. But getting observations from folks, it's a lot. It takes a lot of time and effort to get sample kits to people and receive those kits back. So when people just walk around in the alpine environment and see pink snow, they can just report those observations with this new app that we developed. And you can get it on for Apple iPhones and also for Android phones, which is exciting. It works on all platforms. And my goal for this coming year is to get 10,000 observations, because why not? If everyone who's going hiking and walking around can tell me where there's pink snow, we can get a huge, robust data set on where we're seeing pink. And that, looking at that change over time is going to be, I think, really important for us making predictions about how the albedo changes and the snowmelt changes what can be expected in the future. We also have one sponsorship. So Hydroscape Stickers is a local Bellingham company, and they support us. Gretchen Leggett, who runs that, is also a volunteer and has done a lot of artwork for us so that we make flyers to hand out. I have stickers that she has developed for us as part of the outreach for Hydroscape Stickers, which is great because when people volunteer, they want a cool sticker. So all of these things help continue to engage your volunteers. I want to take a few, when you're doing community outreach projects like the Living Snow Project, one of the, it's great to say, yeah, we've engaged people, I have hundreds of people, we have had over 350 people sign up to volunteer. I don't have stats on exactly how many of those folks have actually collected samples and then returned them to us. We're still working on optimizing that whole process so that we can improve our efficiency. But we're also interested in what the impact of volunteering has on the participants. And that's because this idea of science is also engaging in mentorship and stewardship. So those things all should come together, because if we're interested, yes, these snow algae are potentially endangered or threatened at least because their ecosystem is going away. What does participating in research about that do for motivating behavioral changes? Because really that's what we all are talking about right now. We don't have to demonstrate that climate change is having an impact, but what do we do about it? So the idea that science leads to knowledge, which leads to action, exists, but we wanted to actually see what our role in that could be, creating a participation opportunity in science. And so to do this, I'm collaborating with Dr. Rachel Severson, who is at the University of Montana in the Department of Psychology, and she and then two graduate students, Callie Coffey and Phoebe Bean, are working with me, and we developed a survey that we're giving to volunteers to understand the impact of these science-based experiences on people's understanding of nature and inclination towards conservation. And so this was the introduction to the survey. It's evaluating environmental attitudes, connectedness to nature, and perception of science and conservation by participating in the Living Snow Project. And so folks were asked a couple of questions and did a little interactive game. And so we had 40 respondents to our pre-survey, and we're still accepting responses from our post-survey, so that closes in a week. So I don't have any data to show you, but this is another way in which, as a scientist, I'm a natural scientist, not a social scientist, but I'm collaborating with social scientists to understand the impact of these citizen scientists on conservation. We have some other planned events as well, so we want to do some storytelling events, because people who are collecting samples for us are collecting them when they're going out and having an adventure in the mountains. So they're either going backcountry skiing, or they're going to climb a mountain, or they're going on a hike. And usually when people do those things, they have some kind of adventure. Sometimes they have an epic, sometimes they have a funny story. People who do those things love to share stories with each other. So we'd like to hold a storytelling event to help build a community of people who are participating, so that we can think about it being fun. At the same time, we think about doing volunteer work. And then we have a story mapping project that is being led by Jack Gavin in my lab, who is a Huxley student. And here's Jack collecting a bunch of snow algae this summer, and he created a story map from one of our volunteers' adventures. And this is the URL, which I can share on my website if folks want to go and see it. And if you scroll down in the story map using the Esri tools that Jack has learned how to use in his GIS program, you can see both where the tracks of where Paxton went skiing, what he was thinking about, all of the different fun things that come along with telling the stories of an adventure, in addition to the fact that he actually just collected a sample. Okay, and so the last thing that I was going to talk about today is outreach. So Living Snow Project contributes to my research into helping us build robust data sets. We're learning about how this kind of thing might impact people's inclination towards conservation. And then we also outreach by working with other community programs. So we work with a program called Snow School in collaboration with a bunch of different groups and other education programs, and also with the media. And so Snow School is a partnership between the Mount Baker Ski Area, the U.S. Forest Service, the Northwest Avalanche Center, Winter Wildlands Alliance, the North Cascades Institute. Somehow I missed the, it's because I took this off of it. The North Cascades Institute is actually here, and they're going to be available to talk about being Snow School instructors after my talk, and Western. And so all of these folks are working together to run a program where we actually get to do all of these things at once. We get to teach kids about science. We do the mentorship because the instructors for Snow School are students like you that are teaching then middle school students. And stewardship, so we talk about stewardship, water, watersheds, the impact of climate change on snowpack. And so students who come to this program get to learn about analyzing snowpack with the Northwest Avalanche Center, and then they get to learn about how we sample for snow microbes and how we do science on snow microbes at a station that my lab has developed called Snow as Habitat. And it's really fun. They also get to learn about navigation, and they get to go run around and play in the snow. Many of those students who participate in Snow School haven't ever been to the mountains before. And so this really gives them this comprehensive picture about how when we move up the watershed, the snowpack in the mountains is related to the river flow, is related to the bay. So they get to see themselves within that landscape, but then they also get to learn about science, which is primarily the Living Snow Project's role. So we allow the students to help us test hypotheses. So remember I showed you that, talked about how the algae are dispersed and spread around. We can talk about testing these different hypotheses with students, because if we expect the swimming hypothesis, we would sample for the algae deep in a snow pit. If we expected the wind dispersal hypothesis, we could sample from the top if it was being dispersed with wind or through precipitation. And then again, we get to students are teaching other students, so there's multiple layers of mentorship within the school Snow School program. I've also worked with a number of different print and media over the last few years to help spread the word about Living Snow Project and about the results that we have when we're studying these snow microbial communities. So there's a couple of different articles. We've been in the Seattle Times a few times. We were in King 5 News 2 springs ago. And then just this past summer, I did some work with Oregon Public Television for a show called Oregon Field Guide, and there's going to be a show that I think it's going to be in January that it'll be out, that talks about the Living Snow Project and snow algae and all the research that we do in my lab. This is, there's Jack again, and Emily McGinty, who is a recent Huxley graduate. And then I also went out with Sarah Hoffman from Seattle Public Television, who's doing a story on scientists and their connection to their work. And so there's a lot of ways in which I spend a lot of time with these different media entities. Not so much time that it's overwhelming, but it's important to invest time to both communicate the science that I'm doing and also work to continue to engage more volunteers. All right, so that is about my time. I put up my email, so if you have any questions, you can email me. You can also check us out on Instagram, where I try to post regularly for what we're doing. Before everybody leaves, Snow School is recruiting volunteers. 2 kinds of volunteers. One, people who work at the snow algae station and are interested in talking about the biology, and also general Snow School instructors who help the students kind of come up and move around from stations. Volunteers are students like you and folks that volunteer because the ski area supports this project, and it happens at the ski area. It's unpaid, but you do receive vouchers for lift tickets for each of the days that you volunteer, which is a little bit of an incentive. So students who are interested in hearing more about being a Snow School instructor, just stay put, because the North Cascades Institute folks are going to tell you a little bit more about that when we're done. And with that, I have a few minutes to answer questions if people have questions. Okay.
gotten to name any of them yet?
No. So in order to actually name the species, we have to grow them in culture and characterize other things about their biology, which is really, really hard. So even though I know they're new species, I haven't been able to name them.
Spring and summer giving locations?
Yes, we did that a little bit last year, but this year I'm going to be really specific. I want to get weekly samples from Glacier Peak. Glacier Peak is an area where we get huge snow algae blooms. We think it's really interesting. When I look at the community structure, we have transects that go up the mountain and it's different at each elevation. It's also super hard to get to. So it is the kind of place where I definitely could not go every week. Yeah.
So you talked about what dispersal is getting have you looked at like link backs to say like what those tracks could be from?
That is a really great question, and there was actually a paper just published last year that did that with bacteria that grow in snow. I have tried. It is a bit complicated when it gets down to the basin scale because wind patterns in a place like Bagley Basin, where we sample weekly, it can be all over the place. And so that is a research objective for the future that we're writing into grant proposals, where we can actually just measure the wind patterns within a particular area and then relate that. So overall, like in a general sense, like looking at where the jet stream is moving around and then linking that to where we see algae relatedness across like larger scale is something that I've tried to do, but it's really difficult. It's a great idea. Well, that's a great question. They're the primary producers for the snow, and that schematic I had showed that the fungi eat the snow algae sugar, and there's little animals that live in the snow, so rotifers. You don't know what rotifers are, they're freshwater. Little animals, they actually eat the algae. I have some pictures of rotifers eating the algae. And then other insects eat a combination of those other microbes that are in the snow. And then there's there's insects that eat other insects that are often eaten by birds. So there is a simple but multi-level food web that exists within the snow.
Do you think it's similar to like a marine food web where it's like bottom up versus top down and independent? So like bottom up would be like the snow algae, have more influence over the bottom.
Yeah, I think the algae do have more influence. Yes, and that is I'm in the throes of writing a proposal right now to study the physiology of the algae in relation to different snow chemistry, so that we can understand like how does the availability of nitrogen and phosphorus and things in the snow impact the physiology of the organisms.
Have you looked at how avalanches affect the community?
That is a really great question, and I haven't ever thought about it, except for this past year I went to New Zealand to run a study very similar to what we've done in the Cascades, where we sample across the New Zealand Alps. And snow algae are much harder to find in New Zealand than in the Cascades, which was a bit of a surprise. And commonly, we were sampling old avalanche debris, and it made me think that there could be a relationship. And in Washington, we have so much snow algae that typically I avoid avalanche debris as a place to sample, because it's obviously transporting snow algae downslope. So up to this point, I've been avoiding it, but because I just had this experience in New Zealand where we were sampling it, we started to think about how that might be playing a role in dispersal, or at least moving populations around in a basin scale. Yeah, that's a great idea.
Okay, so let's transition to another talk on the snow school. And if people need to sneak out quietly, they can, but it's worth sticking around if you can.